Welcome viewers. Today we are going to have a discussion on sterilization techniques. Before starting our discussion, we will be going through certain important terminologies. Let's start with sterilization. Sterilization is derived from a Latin word called sterilis, which means barren or unable to produce offsprings. It is a process by which living cells like microbes, bacteria, fungi, prions, endospores are removed completely from a particular object. An object is sterile means it's completely free from all sorts of viable organisms.
Ma'am, we have heard of a term called disinfection.
Yeah.
What is the basic difference between disinfection and sterilization?
Actually, in disinfection, we are targeting only those microbes which are responsible for causing diseases, or we may be calling them pathogens. So, in disinfection, we are removing only the pathogens. But disinfection is not sterilization. It means there might be certain other microbes still present there, but they are harmless to us.
Okay.
Next terminology that we will be discussing is sanitization. Sanitization is a bit similar to disinfection, but the basic difference between sanitization and disinfection is that in sanitization we are also targeting the pathogenic bacteria, but we are not completely killing all the pathogens, but we are decreasing their titer to such a value that it is tolerable by the human beings.
What is the basic difference between antisepsis and sanitization?
Actually, sanitization and antisepsis basically are same, that in both the methods we are killing the pathogenic organisms and we are decreasing their titer. But the thing is, sanitization is mostly applicable on inanimate objects. For example, if you want to clean this place or this tabletop, then you will be using an agent called sanitizer. But if you want to sterilize your tissues, then you will be using a chemical compound, then it will be named as antisepsis. Right? So, the importance of sterilization or antimicrobial control agent study is of immense practical importance from the viewpoint of human health. Though we all know that microbes are helpful to us, most of them are beneficial, but still there are certain microbes which are responsible for causing diseases. On the other hand, there are certain microbes which are responsible for spoilage of food. So the prime target of sterilization or antimicrobial agent will be, number one, to target those pathogens which are responsible for causing disease, and number two, to kill or reduce the titer of those microorganisms which are responsible for spoilage of food or for contamination of water or fruit juices, etc. I will be discussing with you the different factors on which antimicrobial agents are depending. So, number one factor is called as the population size. Population size means as soon as the microbial titer increases, obviously the duration of the time needed to combat those microorganisms will increase. So, number one factor is population size. Number two factor is nature of the population. If the nature of the population is so that it comprises of some newly cultured cells which are easy to kill, then the exposure time for antimicrobial agent will decrease. On the other hand, if we are dealing with older cultures, then it will need more time to kill those microbes. In a similar way, we all know that bacteria can exist in two forms, vegetative cells or spores. Spores are comparatively difficult to combat. So, if in a population the concentration of spores are high, then the duration of exposure will increase. So, the third factor on which the antimicrobial agent's efficiency depends is the concentration or intensity of the antimicrobial agent. We all know as we increase the concentration of antimicrobial agent, their killing efficiency increases. But there are still certain exceptions. As for example, 70% alcohol is a better disinfectant or is a better antiseptic as compared to 100% alcohol. Here we can see that decreasing the alcohol concentration by adding water has increased the effectiveness of alcohol's killing efficiency. And next factor on which the antimicrobial efficiency depends is the temperature of application. Here we will see as soon as the temperature is increased, the time needed to kill the microbe will decrease. And finally, it again depends on environmental factors. Say for example, you want to kill a microbe which is residing in an environment, that environment is favorable for its existence. Then you might be needing some excess harsh methods to combat that pathogen. Or in other words, if the bacteria or the pathogen is residing in an unfavorable condition, then the antimicrobial agent needed to combat the pathogen will be milder enough to kill it. So precisely, if we try to focus on the topic of sterilization or killing efficiency of a bacteria or a pathogen, we will find that there are in all 3 different ways of making an antimicrobial agent. Number 1 are physical agents, number 2 chemical agents, and number 3 is antimicrobial agents. I would like to pay tribute to those eminent scientists who have done enormous efforts in achieving these targets. So first name that comes in our mind is Louis Pasteur. Louis Pasteur was the first scientist to discover that the diseases are caused by agents he called them germs or bacteria. To combat this bacteria, he designed a method called pasteurization, wherein he could increase the preservative capability of wines. Later on, these methods were used for the preservation of milk. Next scientist whose name comes to our mind is Chamberlain. Chamberlain first developed the concept of filter sterilization, wherein we can sterilize any solution and make the solution free of any type of bacteria. Next, the scientist called Joseph Lister bring an enormous change in the lifestyle of human beings by introducing the concept of antiseptic surgery. Therein, what he did, he started using carbolic acid and spraying the carbolic acid in the operation theatres, drastically reduced the death rates in the post-operative cases. Now we will be coming into the various types of physical agents depending on which sterilization can be done. So different physical agents comprises of heat, radiation, filtration and low temperature. In today's lecture, we will be focusing basically on the role of heat and low temperature in sterilization. All the microbes do have a definite temperature requirements for their growth. If we exceed the temperature above that temperature, then it becomes difficult for them to survive. Usually, heat destroys the nucleic acids and proteins by disrupting their normal textures. Moreover, what happens that heat is detrimental because it drives off water from the body of the microbes, and then it becomes fatal for that bacteria.
On what factors the killing rate of heat depends?
Killing rate of heat depends on 2 factors, time and temperature. For example, if we are dealing with the killing of a pathogen called Mycobacterium tuberculosis, the causative organism of TB, we will see that it requires 30 minutes to kill the microbe if the temperature is 58 degrees centigrade. But if we increase the temperature from 58 degree to 65 degrees centigrade, then the time reduces from 30 minutes to 5 minutes. On the other hand, if we further increase the temperature to 72 degrees centigrades, then it takes only few seconds to combat the entire microbial population. Next, we are coming to the types of heat used as a sterilizing agent. So, heat can again be divided into dry heat sterilization or moist heat sterilization. First of all, we will take up dry heat sterilization. It can be further divided into two different parts. One is called incineration and number two is called the use of dry heat in hot air ovens. So first of all, we will take up the use of incineration. You all know that in our microbiology labs, we directly flame the inoculation needle to remove all the microbes that are present on the needle. So it is very much essential that the needles should be incinerated properly before inoculating any culture directly into a sterile media. Right? So Bunsen burner has got the temperature so high that is sufficient to kill all the microbes present there. We are using incineration in hospitals wherein disposable gowns or OT usage things are destroyed first by incineration, then only they are discarded into the environment. Next, we will see the use of dry heat in case of hot air oven. Hot air oven actually uses dry heat for sterilization, but we all know that the penetrance power of dry heat is very less. That is the reason we need very high temperature and a longer exposure time to carry on the complete sterilization or removal of viable cells as well as endospores in hot air ovens. Here what happens that this dry heat actually carries on the oxidation of all the proteins, right? And this oxidized proteins loses its efficiency, as a result of which we all know that most of the enzymes within our system are proteins in nature. So because of this high oxidation rate of the proteins, the enzymes get inactivated and microbes die off. Moreover, this heat generates an arid environment which leads to the internal burning of the cells or the microbes. So dry heat sterilization needs a temperature of 160 degrees centigrade and a duration of 2 hours is supposed to be enough to get rid of endospores. But the thing is, it has got certain limitations, because such higher temperatures might not be tolerable by the other agents like growth media particles. This cannot be sterilized within a hot air oven. So precisely the things that can be sterilized in a hot air oven should be dry in nature, like glasswares, surgical instruments, petri dishes, conical flasks. These things can be sterilized properly in hot air ovens. Right? And one benefit of dry heat over moist heat sterilization is that there are certain surgical instruments which become dull in nature, or they lose their luster, or they may gather rust in moist heat sterilization. These type of problems are not faced during dry heat sterilization. Moist heat penetrates more easily as compared to dry heat. Reason behind that, it is the water molecules which can conduct heat better than air. The method by which moist heat carries on microbial killing is destruction of nucleic acids, denaturation of proteins or enzymes, and in many times it leads to disruption of the cell membranes. Now, moist heat sterilization can again be of different types. It can be boiling of water. Number 2, it can be autoclaving. Number 3, it can be pasteurization, or it can be partial sterilization, or we may call it as tyndallization. So first of all, we will be starting with boiling of water. It has been a method from ancient days that people know that anything we want to sterilize, if it is boiled in water, then it undergoes perfect sterilization. So 10 minutes of boiling is enough to kill the viable microorganisms. But this time is not sufficient to take care of the endospores. So we need to increase the duration up to 30 minutes. Sterilized steam is used for killing of the microbes. In autoclaving, what we see that the microbes are getting killed at a temperature of 121 degrees centigrades and a pressure of 15 pounds per square inch. The principle of autoclave lies that here we keep on boiling the water, and this boiling water generates a steam. This steam pushes off all the cold water or cold air or the normal air that was present inside the autoclave chamber. Then what is done, that the chamber is closed and the steam inside the autoclave starts increasing in pressure. With increase in pressure, the temperature of the autoclave starts raising, and it is allowed to reach a temperature of 121 degrees centigrades and a pressure of 15 pounds per square inch. In this condition, if we maintain the things to be sterilized for 15 minutes, then all the endospores and viable microorganisms are supposed to be killed. But this duration might be increased if the volume of material to be autoclaved is increased rapidly.
Ma'am, how can we say it with surety that the things inside the autoclave are perfectly sterilized or not?
It is very difficult to understand that whether we have reached exact sterilizing point or not. That's why we have started using certain biological indicators. These biological indicators means we are dealing with the spores of Geobacillus stereothermophilus, which is very much resistant towards heat killing. So if these spores are destroyed during autoclaving, then we can say with immense surety that the process of sterilization has been carried on perfectly. So what is done, a culture tube is taken. Within that culture tube, one ampule containing a sterile media and a paper which is dipped with the spores of Geobacillus stereothermophilus is kept inside the autoclave. After the completion of autoclaving process, what is done, that this ampule is broken aseptically and it is allowed to grow in an incubator shaker for definite period of time. If we see that there is no growth of bacteria, then we can say that obviously all the spores have been killed during autoclaving, and it means that the entire process that was being carried out during the autoclaving process was a perfect one. Okay. The things that can be sterilized in autoclaving are mostly, these are used in hospitals and laboratories. It is used for the sterilization of the growth media for the microbes. It is used for the sterilization of surgical instruments. It is used for sterilizing various glasswares. It can be used for sterilizing blankets, operation theatre used labwares, and many more things. The application of autoclave is really endless. But as we all know that every process has got certain limitations, right? Similar is the case with autoclave. Therein we can see that autoclave is unable to kill prions, right? Another limitation with autoclave is that as moist heat is the principle for killing of the microorganism, this moist heat sometimes leads to deposition of rusts on certain metallic surgical instruments. That's why we have got certain limitations in its usage. To overcome this thing, we have developed another type of autoclaving that we call as pre-vacuum autoclave. Therein what is done, that a separate instrument is used to take out the cold air which was present inside the autoclave. It is taken out. Then what is done, here the pressure that is applied is pretty high as compared to 15 pounds per square inch. Here the pressure is taken up to I think 34 pounds per square inch, and the temperature is also raised drastically high. So in this tremendous pressure and the higher temperature that we have reached, the duration of autoclaving is reduced from 15 minutes or half an hour to say 4 minutes. So as the things which are getting autoclaved are residing inside that condition for just 4 minutes, so the probability of getting the disadvantages of autoclaving, just as developing of rust, is lost. And number two, that the prions which were not being killed during normal autoclaves are killed in pre-vacuum autoclaves. So after the completion of the autoclaving process, the steam is taken out with the help of some other suction methods. So the steam never remains inside the autoclave for a longer duration of time.
What is the advantage of a pre-vacuum autoclave over a simple autoclave?
So the first difference with benefit of pre-vacuum autoclave is it is being used for a shorter duration of time. So the detrimental effect, as I said, that developing of rust or the autoclaved things gather moisture, these problems are there with normal autoclave. It is not there with pre-vacuum autoclaves because we take out the steam after the process of autoclaving is done. And the most beneficial effect is that we are able to combat the prions which were not being killed with the normal autoclave. Fractional sterilization or tyndallization. This method was developed by John Tyndall. In this method, steam is allowed to sterilize liquid compounds or other objects, but this process is carried out in 3 different days. In the first day, we carry out the steaming process at 100 degrees centigrades, followed by incubation of the sterilized material. In this process, what happens during first course of sterilization, all the viable microorganisms are killed. Only the spores are the ones that escape killing. Then what happens, that we are allowing this sterilized material to grow under normal temperature. Under these conditions, the spores get favorable conditions for germination and they are then converted into vegetative cells. During the next day's sterilization, they get killed. So this process is continued in 3 different days. That's why we call it as fractional sterilization or tyndallization. This method is very nice as we have taken measures to kill all probable varieties of spores present inside the bacterial suspension. It is developed by the famous scientist Louis Pasteur. This method was initially used for decontamination of wine. Nowadays, it is used for sterilization of milk. This can be of 3 different varieties. First method is called holding method, second is called flash pasteurization, and third is called ultra high pasteurization. In holding method, the temperature is 62.9 degrees centigrades and the process is carried out till 30 minutes. In case of flash pasteurization, the temperature is 71.6 degrees centigrades and here the process is carried out for 15 seconds. And finally, we have got ultra pasteurization where the temperature is increased to 82 degrees centigrades and the duration has decreased to 3 seconds. So pasteurization is basically not sterilization. It is just killing of viable microbes. We are not taking care of the endospores, right? At this temperature, we are killing only the pathogenic microbes, but the thermophiles still remain into the system. But we are not worried about them because thermophilic bacteria are unable to grow inside the human body, where the temperature is pretty less, which cannot support the growth of thermophiles.
Ma'am, how is ultra high temperature method
Yeah.
preferred over pasteurization?
In case of pasteurization, we are unable to kill all the microbes. We have taken care of only the pathogens. We have not killed the endospores. But in the ultra high method, here we are increasing the temperature enormously, as a result of which the spores are being killed and the other microbes are also killed. And we have taken some precautionary measures to keep the food value intact. Many a times we prefer to restrict the growth of microorganisms by freezing them in or by refrigeration. Here what happens that we are refrigerating it at -30 or -70 degrees centigrades. These methods are used for maintenance of bacterial cultures. Here what happens that as the things are being frozen or water is being taken out from them, the cells stop replicating, they stop reproducing. But they are still viable. They are not completely killed. So these type of refrigeration methods are mostly used in food industries for preserving food materials and foodstuffs that are to be exported from one place to other. But there is a drawback, as we have said, that we have not killed the microbes. They are viable but in an inactive stage. That's why whenever we want to consume these frozen products, it should be first cooled down inside the refrigerator and should be consumed immediately. Otherwise, there are chances of them becoming active and imposing hazardous effects. So, in this lecture, we have discussed about the importance of sterilization, and we have discussed the various means of sterilization, giving precise focus on different physical agents, and we have discussed elaborately about heat as a sterilizing agent and low temperature for sterilization purpose. Thank you.